This is the first of probably a couple of prep videos for lecture 9. We're going to talk about single gene inheritance. So we've gone through kind of the structure of DNA, how nucleotide changes lead to changes in proteins, and therefore give rise to phenotypes. And now we're going to, and then we talked in class about both mitosis and meiosis. Meiosis is the division that we focus on in genetics because it's the division that leads to the gametes, which are passed on to offspring. And now we're going to start looking at single gene inheritance. And when you do this, please pay attention to how the alleles, the variant forms of the genes, move to the next generation, and try to make a connection between that and the movement of the chromosomes during meiosis. And that will, I think, help you make a good, strong connection and understanding of this topic. I know for most of you it's already been covered in other courses, maybe even in high school in some cases, but we'll go through it in a fairly simple way, and again, do try to make the connection with the movement of chromosomes during meiosis. That will help. And there will be, of course, some open access resources on your Codon site. There, you can also access the textbooks that we've put aside in the library. Any section that describes Mendel's first law and Punnett squares and how to use them will be sufficient. If the text is a good text, it should explain this topic. Okay, so Mendel's achievement was that he was able to describe the inheritance of alleles, which are, as I said, variant forms of genes, so genes with copies of genes with slight nucleotide differences. And he could describe this based on the segregation of chromosomes, which is why I asked you to try to make that connection. But he was able to do this and describe the process without even knowing what the units of heredity that he was working on were. So he called them units of heredity because he didn't know exactly what they were. Nobody knew about DNA and at that time. And at the time, a lot of the way people thought about heredity was that it works somehow by blending. So somehow, you've got two different parents that have two different phenotypes, and the offspring that are produced are somehow intermediate between them. The red of the flower petals and the white of the flower petals blend somehow to produce, the pink. And there are definitely cases of this kind of what looks like blending inheritance. And you often, may have a very tall parent and a very short parent, and, the kids they have are somewhere in between. And so that was the idea that people had, but they weren't necessarily basing it on careful, very precise observation of crossing one particular individual to another and then following the outcomes. And that's one of the keys to Mendel's success. He was trained in physics, actually, and so he had a focus on quantifying things, which a lot of people looking at the biological world weren't really doing, counting and assigning numbers to things. And he also had very careful ideas of controls and sort of, how would you design your experiment properly. And so he brought that kind of training to this field, and it worked out very well. So, as I said, we're just going to look at the first law. We'll talk about the others later on. But so the things that made his experiment work were that he chose the right experimental organism. If he had been working with, he worked in a monastery, and so they had livestock as well as different crops and so on. And if he had chosen any of a number of other organisms, especially, sheep or goats or something like that, he would never have been able to get the numerical results that he had. They take so long to reproduce. He also selected phenotypes that had discrete phenotypes, so they weren't sort of varying shades of flowers. It was either purple flowers or white, and all the white ones looked exactly the same, and all the purple ones looked exactly the same. And they were easy to tell. You didn't have anything where you weren't quite sure what the flower color was, or what the pea color was, and so on, the pod color. And he chose characters that had only two different phenotypes, right? Then he designed his experiments very carefully, and I'll talk about that a little bit. He quantified the data, and this is really, really important, the actual counting of the phenotypic classes, and then using those numbers to try to sort out and understand what was happening. He came up with a hypothesis, he made predictions, and then designed experiments to test those. So he did an experiment, he got some data, he thought, what would explain this? Okay, here's my hypothesis, and then if I'm right, what should I see if I do the following additional experiments? And he tested those hypotheses that way. Now, the peas were a very good experimental organism. They grow really easily. They have lots of progeny. So if you think of a single pea plant and it has all these different pods on it, and each pod has maybe 8 to 10 peas inside, and each of the peas is one offspring of a cross. And so, you know, even on this branch, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So say 80 offspring right there. That's a really good number. Oh, not 80, 88, because there's another one. And this is just one little part of a plant. You know, you could get hundreds of offspring from a single plant. And they also had, they have flowers that have both male and female parts, and he was able to control the crosses. That is, he could allow them to self-pollinate, which peas mostly do, or he could remove the male parts and use a brush to introduce the pollen of choice to the female part that was left behind on the flower. And so he had the ability to control the crosses in a couple of different ways. And you'll see towards the end why that's important. Oh, I must remember to update this. I forgot to change that. So his luck was he chose the right organism. And it turns out he chose 7 characters, and each of them had the 2 distinct variants for the experiments. Each phenotype was easy to categorize with, and there were no intermediates. And it turned out, very luckily, when he started looking at the characteristics in pairs, they were all on different chromosomes. So peas have 7 pairs of chromosomes, and the 7 characters, there was 1 on each chromosome, which was incredibly lucky, because it would have been really awkward if you know, if there happened to be 2 genes that he was interested in on 2 different chromosomes. One other thing that's very important, many plants are polyploid. That is, they're not diploid. They don't just have 2 of each chromosome. The bread wheat that you know we use to make our bread and so on is hexaploid. There are 6 of each. And if he had decided to work on wheat, we would still not know the results of those experiments, because the mutations that he looked at were all recessive mutations, and you'd have had to make all 6 copies mutant in order to see a phenotype. And this just shows you what they are. Round versus wrinkled seeds when they ripened, and then yellow versus green seeds, separate from yellow or green pods. The wild type is on this side, and the mutant is on the other side. Purple versus white flowers, an inflated pod or a pinched pod. So when the pods ripen, if the tissue is very stiff, the pod will just look full. You won't see the individual piece. But if they're pinched, they kind of shrink down around, and you can actually see the outlines of the piece. Axial flowers, which is the normal, or just terminal, so only on the tip, the growing tip of the plant. And then long versus short stems. Some plants grow to different heights depending on soil nutrition and so on. But there are some plants that are determinate, and that is they grow to a certain height and they stop. And in this case, he could distinguish very clearly tall versus short plants. So those were the lucky aspects of his work. The skill was his physics training, his careful quantifying of the results, the idea of counting everything and calculating ratios, and not just counting 20 offspring, but counting 6,000 offspring and seeing what the ratios were. And then careful design of his experiments based on his hypotheses. And one of the most important things he did was to start with strains that are pure breeding. What that means is you have a plant that's tall, right? And you collect all its progeny and you grow the progeny and they're all tall. And then all of their progeny are tall and all of their progeny are tall. And he had to keep breeding them for many, many generations, several years of work, until he was able to obtain strains that were pure breeding. We now know that they were homozygous, and we'll talk about what that means in a few minutes. But he called them pure breeding because, of course, he didn't know about homozygous or heterozygous genes hadn't even been discovered yet. And then he used those strains, the pure breeding strains, in a series of reciprocal crosses. Reciprocal crosses means, for example, if he crossed a tall by a short, and he used the female part of the tall plant and the male part of the short plant, and then he did it the other way. So he also did a short female cross to a tall male. Because you'll see in a lecture coming up that in some cases it does matter which parent is the female and which is the male. And then he used single characters first, and that's what we're going to talk about now. And then later on we'll talk about when he combined additional characters. So this is just an example, but he did this for each of the pure breeding pairs of strains. And this is just the yellow versus the green, yellow versus the green peas. And so he had a pure breeding yellow line and a pure breeding green line, and he crossed them to each other. And then when he looked at the progeny, all of the progeny were yellow, like 100% yellow. So that was the first result. And then we crossed the tall to the short, all of the progeny were tall, and so on. And we'll see a table of that in a minute. I want to draw your attention to these terms because these are really important. The P generation is the first cross that is made between, in this case, homozygous or pure breeding, yellow and green or whatever. So that's called the parental generation. And then their offspring are called F1. That's the first filial generation. And then if these are crossed to each other, the next generation would be F2. And if you went further, it would be F3. So this is just terminology. It's very unlikely we'll go to F3 in any of the crosses we cover in the class or that we ask you to work on for practice. But we could go up to F2. Okay. And I think that's all you need to know for this particular slide. But I'll just remind you about loss of function mutations. So what's happening here? What happened to the green trait, right? And just remember what I talked about, lecture 6. There was a little additional bit that I talked about, loss of function mutations. In a loss of function mutation, the protein product doesn't work, or it's not produced, or it just doesn't do the job very well. And what that means, though, is that is usually a recessive mutation. It doesn't show up in individuals that still have one good copy. So if you still have a good copy of the gene, you have enough functional protein for wild type phenotype, and it turns out that yellow color is the wild type for this kind of P, right? So these types of mutations are usually recessive, okay? And the mutant strains that Mendel worked with, they were all loss of function mutations. So the mutant ones are going to be the recessive ones. And this is just a reminder. I know you've seen something like this, but this is just a schematic. So the way we denote the genotypes is by just using, most of the time, single letters, but for some, I'll show you different ways of writing genotypes in other examples. But in the case of these plants, it was just a single letter designation, and capital Y is for the yellow color, and that is the wild type, the dominant one. Both of the copies of the gene are being transcribed and then translated to produce a functional protein that leads to that yellow pigment in the peas. There's a mutant copy of the gene that is not producing a functional protein, so there's something wrong with it. The protein doesn't work, or maybe it's not even being transcribed properly. Maybe it's, there's no promoter or something, but you still have one good copy of the gene, and this is how we write the genotype. So this tells us both copies have the dominant allele. This tells us, which you can't see very well, that you have one good copy and one mutant copy. Whenever you're writing a genotype, if the letters are like the Y here, where it could be difficult for us to see by your writing whether you've made 2 uppercase, 2 lowercase, or so on, just put a little bar over the lowercase one, and that way we know for sure what you're telling us. This happens a lot with like O. Is this just I made a mistake, or is it really a lowercase? Well, if I put this little bar over it, then you know that's heterozygous. Same with C. Excuse me. You just make sure it's clear. It's about part of getting the grades is clearly communicating your understanding. And it's too easy to just write something, where the size difference excuse me, where the size difference isn't very much. And it could be that you wrote it homozygous or heterozygous, and we just don't know, right? And then same for this. If both copies are mutated now, this is how we write the genotype, and we now have no functional protein. And so the color is green instead of yellow. So as long as you have one wild type copy, you're going to have the wild type color, but when both copies are mutated, you're not going to have any functional protein. Therefore, you are not going to have the proper yellow color. You're going to have green, in fact. Okay, so that was the first thing. He determined that the green color seems to have gone. So, and that was of interest. So what happens if you allow the yellow ones to self-pollinate? If you do that and you look in the next generation, you can see the green reappears. The green reappears, and there's more of the dominant ones than the recessive ones. In fact, it's a 3-to-1 ratio. If you take those F1, note these are the F1, not the pure breeding, but the progeny of the pure breeding. If you take those F1 yellow and you pollinate with green, or you take the green and you pollinate with the F1 yellow, you can do the cross either way. And when you do, you find that you have equal numbers of yellow and green. So key points here. Is that in both of these kinds of crosses, you see the reappearance of the green trait. So the recessive trait was not destroyed or lost or blended or swamped out by the dominant trait. It comes back completely intact and just as green as the original parent. And we see these very distinctive ratios. Equal numbers of the two in this kind of cross, and then a 3-to-1 ratio in this kind of cross. And we'll talk about the types of, this is called a monohybrid cross. And this is called a monohybrid test cross. That's just terminology, and we'll cover this in more detail in the next recording. But just be aware. This is a summary table, so I'm not going to go through any of this in super detail, but the key things to notice is that um, one of the traits appeared in all of the F1 in each of those 7 crosses. And so that is the one that's referred to as dominant. The other variant is called recessive. Okay. And then secondly, when you allowed these to self, so the F2 here is from F1 cross to F1. This isn't the other cross that I talked about. So you allow these guys to self-pollinate, and you get these ratios. Now notice they're not perfect 3 to 1, even in some case with thousands of progeny, but it's an approximate 3 to 1 ratio. And it's always the dominant trait that's the 3 and the recessive trait that's the 1. And, and that ratio helped Mendel to explain what he thought was going on. And so these are those key observations again. You know, the dominant trait shows up in the F1. You have the recessive phenotype showing back up in the F2 generation. So it had to have been present. It just wasn't seen. So it didn't get lost. It didn't get blended or anything like that. And the results of crosses in both directions. So the reciprocal crosses always gave the same result. So it didn't matter which was the female part of the plant and which was the male part of the plant. You still got the same 3 to 1 ratios. You still got, you know, the dominant trait showing up in the F1 and that sort of thing. Okay, and I'm going to start here on the next recording. So those are the results, and then we're going to launch into how do we explain them, and then how do we describe them and the proper ways to write out crosses and that sort of thing.